everyone, welcome to lesson 4.2 on Punnett squares and probability. Today's lesson and focus will be on monohybrid crosses, and next lesson we will go on to dihybrid crosses. Now what's really important for Unit 4 is as we go through these lessons, please make sure to take good notes, okay? So in class you'll be doing a practice worksheet which is going to cover the examples mentioned in this video. So if you want to write your notes, type them, print them out, write on that, all of that's completely fine, but you need to be able to access them and you need to understand what you actually did. So writing down the steps and maybe extra notes for yourself is highly recommended. So we're going to go ahead and start off with a video. Okay, so pause here, click that link, answer these questions, and then come back whenever you are finished. Okay, so we should now have those questions answered. So Gregor Mendel was the father of genetics, and we know that genetics is the study of heredity. Fertilization is the joining of male and female gametes to produce a new cell. So peas are usually self-pollinating, which can lead to what we call true breeding plants. And Mendel used cross-pollination of true breeding. Okay, and true breeding again is just that self-pollinating plants, and they're always going to produce offspring identical to themselves. So Mendel used cross-pollination of these plants to produce new combinations and study the results. And then a trait is a specific characteristics of an individual. So some vocabulary over the next couple slides we're going to get to. So the first thing is genes and alleles. So an individual's characteristics are determined by factors that are passed from one generation to the next. A gene is a sequence of DNA that codes for a protein and thus determines a trait. And that can also be a factor that's passed from a parent to offspring. So for example, there is a gene that determines the trait for hair color. An allele is the different form of a gene. So an example of that is the possible alleles for the gene for hair color include brown, black, blonde, red, etc. And then we also have these terms dominant and recessive, which you've probably heard before. So some alleles are dominant and others are recessive. For something to be dominant, an organism with one dominant allele will exhibit the dominant form of the trait. Recessive is an organism must have two copies of the recessive allele to exhibit the recessive form of the trait. So, first check for understanding. Pause here, read this, and try this on your own. Alright, so first check yourself. Okay, so for number one, the gene would be the pea seed pod. Okay, and then the allele would be the two possible things or appearances of the pea seed pod. So that would be smooth or constricted. Number two, if a round seeded plant and a wrinkled seeded plant are crossed and all the offspring are round, which allele is dominant, which allele is recessive? So you're not given a whole lot of information from this problem, but because we have this here, this true breeding, okay, then we can make an accurate assumption, which is both are true breeding and all the offspring are round, then the round has to be the dominant trait and wrinkled has to be recessive. And we can conclude that because none of the offspring exhibited the wrinkled allele. So let's talk about Mendel's experiments. Okay, so we're going to have these three terms, P generation, F1, and F2 generation. Okay, so the P generation is what we call parental, and these are true breeding. Okay, again, basically meaning like self-pollinating. F1 is the first filial generation or the offspring of the P generation. And the F2 is the second filial generation or the offspring from the F1 cross. Okay, so for Mendel's experiment, here's a nice image of that. Okay, he had his P generation and F1 here. So his F1 cross are the results of the P, okay, being crossed, F1. All of the offspring ended up looking like one parent from the P generation. So we had round here, yellow, yellow, gray, gray, right? So this whole F1 generation all looked like this parent, okay? And then Mendel also did the F2 cross. Okay, basically he took two of the F1s and crossed those again. Most of the offspring looked like the F1 generation, but approximately 1/4 looked like the second parent from the P generation. So this was really indicative, okay, for Mendel's experiments. Okay, so F1 cross again, just a summary. Here is what he got. Here were his results of the F1 cross, and they all basically looked like one parent from the P generation. And then his F2 cross, okay, approximately 3/4 looked like this parent, okay, that tall axial green, and then 1/4 actually looked like this one, short terminal yellow constricted. So how did the short trait disappear and then reappear in the later generation? And that's how Mendel came up with this law of segregation. So during gamete formation, alleles segregate from each other so that each gamete carries only a single copy of each gene. Okay, so what does that really mean? Well, let's look at this picture, okay? Each F1 plant produces two kinds of gametes, okay? Those with the tall allele and those with the short allele. So we have big T, little t, big T, little t. Okay, the alleles separate during gamete formation and then pair up again in the F2 generation, with the capital letter representing the dominant allele and the lowercase letter representing the recessive allele. And so then the F2 generation had new combinations of alleles. Okay, again, because that's showing how these gametes separate from each other or segregate from each other. And then down here in the F2, that's how we get different and new combinations. So go ahead and pause here, read this, and try this check for understanding. Okay, so identify the generation based on the situations and their descriptions. Okay, so for B, we'll go ahead and do B first because it's the P one. Okay, it says true breeding, yellow seeded plant, and true breeding, green seeded plant. If you see that true breeding, you know that it has to be the P generation, right? That's the only one that had true breeding. Okay. Now let's go back and look at C, two yellow-seeded plants that were the offspring of the two true breeding plants, okay? F1 was the offspring of the P generation. Whenever we see true breeding, we think that is P, okay? And then the last one, yellow and green-seeded offspring resulting from a cross of two yellow-seeded plants. So the F2 generation, that's the only generation that had a green-seeded plant appear. Okay, also this whole two yellow-seeded plants, again, as the parents, that's the only one that had the F1 generation, and F2, again, is just the offspring of that F1. All right, so now let's talk about probability. So probability is the likelihood that a particular event will occur. And so, for example, when you flip a coin, there are two possible outcomes, heads or tails. And the probability of either outcome is theoretically equal. Again, we know that if a coin is heavier on one side or, what have you, conditions, environment could change it, but theoretically it is equal. So the probability that that single coin flip will land heads up is 1 out of 2, 1/2, or 50%. If you were to flip the coin multiple times, then each event is independent. So the probabilities of each event can be multiplied. So the probability of flipping 3 heads in a row would be 1/2 times 1/2 times 1/2. So you have a 1 in 8 chance of flipping heads 3 times in a row. Now what's the probability of flipping 5 heads in a row? So kind of pause here and try this on your own. So the math is pretty much the exact same as the previous example here, but now you just do it 5 times. So 1/2, 1/2, 1/2, half, oops, and you get 1 out of 32 chance of flipping 5 heads in a row. Okay, so segregation probability. Why do we have to go back to probability in the middle of that law of segregation? Mendel's F1 cross produced 1/4 short offspring because both tall plants had one tall allele and one short allele. Okay, so here we're looking at this, right? Each F2 gamete had a 1/2 chance of carrying the short allele, and 2 gametes combined to make offspring. So the chance that each of the offspring would end up with a short allele or 2 short alleles is 1/2 times 1/2, which is a 1/4. Okay, now if you're confused by that, we have another way of looking at this through Punnett squares, which we're going to talk about momentarily. But before we go into that, we have to talk about genotype and phenotype. So individuals who are big T, and big T, little t, may look the same but carry different alleles. So what we have is we have this term homozygous, and that's organisms that have 2 identical alleles for a gene. So 2 big letters would be considered homozygous dominant. 2 little letters or lowercase letters would be considered homozygous recessive. A heterozygous organism is one that has 2 different alleles for a gene. So if you have 1 capital letter and 1 lowercase letter, that would be considered heterozygous. The genotype is the genetic makeup, so literally just the letters, okay, or the coding or the alleles that are in that specific organism. And the phenotype is the physical characteristics that occur because of that genotype. And for example, that would be the pea plant that is short. Okay, so here's the example here, right? This one would be homozygous dominant. This would be homozygous recessive. And then these two would be heterozygous. Now their phenotypes, we have this is a tall plant based on their genotype big T, big T. This is a tall plant based on their genotype, which is heterozygous. However, it still has that dominant trait, so it's going to appear tall. Same thing with this one. And here we have a short plant. It is homozygous recessive, okay, and its phenotype is short. So now we're going to get into some Punnett squares, which you may have done before. However, if you have not, that's perfectly fine. We're going to do some practice. So Punnett squares use mathematical probability to help predict the genotype and phenotype in combinations in genetic crosses. So today's lesson, we look at monohybrid crosses. Okay, so first one, practice. In humans, widow's peaks, we're going to call that H, okay, big H, are dominant to a straight hairline, little h. So a person who is heterozygous could produce two types of gametes. 50% would have a big H, K, or the widow peak, and 50% would have little h, or the straight hairline. And a cross between two people heterozygous for widow's peak, the Punnett square would look like this. Okay, so we have heterozygous, so here's what we're going to do. We're always going to write out the genotypes first. So big H, little h, okay, big H, little h for both parents. They're both heterozygous. Now we have our Punnett square, okay, this block here, 4 squares, and we're going to put the gametes, possible gametes for each parent, okay, at the top of the Punnett square and these sides. So we have big H, little h that we just got from here, okay, and then big H, little h, same thing for parent 2. Then you fill in the Punnett square. Now when filling in, okay, you are going to tape the top parent, okay, and if it has a capital letter, you will always fill it in first. So from this combination, we have a big H and a big H. Okay, this combination, we have a big H and a little h here. This combination, we have our big H, capital letter always first, little h, and our last one, 2 little h's. Okay, so those are our 4 possibilities. Now, we are going to ask you sometimes what is the genotypic ratio of the offspring. So here's how we write that, okay? It is the number of homozygous dominant to the number of heterozygous to the number of homozygous recessive out of the offspring, or out of the possibilities, okay? So we had 1 that was homozygous dominant, so that's where we get this green number. We had 2 that were heterozygous, that's how we get this yellow, and we had 1 that was homozygous recessive, that's how we got that purple. So this would be your genotypic, okay, frequency, or genotypic ratio. Now, same thing, we might also ask you the phenotypic ratio, and that's just the number of dominant to recessive offspring, okay? So how many are going to appear dominant and how many are going to appear recessive? If you recall, dominant includes both homozygous dominant and heterozygous. If you have one allele or just one copy of that big H, it is automatically going to appear dominant. So here we have 3 that appear dominant to 1 that appears recessive. So for our phenotypic ratio for this example, we get 3 to 1. Okay, so we've reached our first check for understanding. So pause here, read through this. We kind of set this up to help you out and guide you. Okay, try this on your own. Don't skip forward. Try it on your own, and then we will come back and discuss it. Okay, so let's look at this example. So the first one, heterozygous for flower color. I used the big P, little p, okay, heterozygous parent 1, and a plant with white flowers. Okay, for white flowers, that would be recessive, so 2 little p's. Okay, we go ahead and put our parent 1 gametes here, parent 2 gametes here. We perform this cross and we end up looking something like this. Again, always put your capital letters first. If your capital and lowercase letters look very similar, then you may want to do something different to the lowercase letter just to differentiate that. So for here, I just made the P's like bolded and a little bit extra big. So from this, our genotypic ratio would be 0. We had 0 combinations of homozygous dominant to 2, because we had 2 heterozygous, to 2, okay, because we had 0 homozygous recessive. Our phenotypic ratio, again, is just the number of dominant to recessive. So for here, we had 2 dominant to 2 recessive. So 2 to 2, or it could reduce that 1 to 1. Now for number 2, what's the probability that the plants described above would have offspring with white flowers? So remember, white flowers have to be 2 little p's, right? Because that is recessive. So for this one, there was 2 out of our 4 total offspring. Okay, so 2 out of 4 would be 1/2 or 50% chance of that pea plant that was heterozygous crossed with a plant that with white flowers would produce offspring with white flowers. All right, so test crosses. So individuals who are homozygous dominant and heterozygous display the same phenotype, right? So for this example, big Y, yellow, and heterozygous big Y, little y, both appear yellow, okay? However, they have different genotypes. So say you have a yellow pod pea plant that has a dominant trait. What could you do to determine if the plant was homozygous dominant or heterozygous if you didn't know its genotype? You only had the phenotype. So you could perform what we call a test cross, and that is if you cross the yellow pea plant with something that you absolutely know is recessive. So for example, a green, okay, little y plant, then from that offspring you could predict the genotype of the parent. So this test cross, we're going to do some examples on that next class, but here kind of basically your 2 options. If you had big Y for your parent, then all of the offspring would have to be yellow, right? It would conceal that recessive gene. But if you had big Y, half the offspring would be yellow and half green. So that brings us to our observed and expected values. Okay, so the observed value is the number that we actually see, and the expected value is the number that we would predict using Mendelian inheritance patterns. So for example, okay, we're going to go ahead and try this first one. Okay, purple flowers are dominant to white flowers. 2 heterozygous parents produced the observed values in the table here. Okay, what would the expected values be? So we're going to go ahead, and I want you to try this on your own before skipping ahead. Okay, so step 1, complete your Punnett square given the scenario from this problem. Step 2, find out that expected dominant percent or ratio and expected recessive percent or ratio, and then multiply that by the ratio or total number of offspring given here. Okay, so for total number, you'd add up the total number of purple and white. All right, so go ahead and try that one out. And then for number 2, similar thing, okay, but now you have a heterozygous parent and homozygous recessive parent. Those are crossed and produce these observed values. Okay, so this is scenario 2. Okay, so same steps, okay, but now you have different parents. So again, pause here, try this out, follow those 3 steps. Okay, so let's go ahead and look at our first example. This one corresponds here. All right, we did that cross. I did not write it on this slide. Okay, and then we find out our expected dominant percentage would be 3/4 dominant, or 75%, and recessive 1/4, or 25%. Okay, now our total offspring was 200, right? So 157 plus 43, that gave us 200. Now you'd multiply that 200 by, for our dominant, purple, okay, 3/4 or 0.75, and our expected value would be 150. For our white, okay, we'd multiply that 200 by 1/4 or 0.25 to give us an expected value of 50, okay? Now the second scenario, similar idea but different parents, therefore we have different percentages. So our dominant would be 1/2 and recessive would also be 1/2. Okay, we still had 200 total offspring. So we would expect to get 100 purple and 100 white. Again, we expect to have even ratios for that. All right, we've reached our exit ticket. Pause here and try these out on your own. Okay, so go ahead and check yourself. 4 coin tosses in a row that will land on tails. So basically just multiply 1/2 4 times. We get 1 out of 16 chance. The genotypes, 2 big letters, homozygous dominant, 1 big letter, 1 lowercase, heterozygous, and 2 lowercase, homozygous recessive. Number 3, okay, determine the genotypic and phenotypic ratio for a pea plant, homozygous dominant, here for flower color, and a white flowered plant. So we know that that would have to be recessive. okay? So you do that cross, you should have gotten something like this. So 0, that were 2 big P's, all 4 were heterozygous, and 0, that were 2 little p's, or recessive. Then your phenotypic ratio would be 4, right? All 4 are pure dominant to 0 recessive. Number 4, it's asking about this question right here. And there was a 0% chance that those parents could have offspring with white flowers because they are all heterozygous, therefore none of them will appear white. Number 5, so unicorns can be either pink or purple, but for a unicorn to be purple, at least one of its parents also have to be purple. So purple would have to be dominant, and this can be kind of confusing, but think of it like this. If we had 2 pink recessive unicorns and had offspring, there's a 0% chance that they will ever have a purple offspring. However, if 2 purple unicorns had offspring, there is still a chance that they could produce a pink unicorn if those purple parents were heterozygous, okay? And then the last one, so for number 5 above, determine the genotypic and phenotypic ratio if 2 pink unicorns have offspring. So basically you should have just done a cross with 2 recessive parents, so 2 little p's for both. Your genotypic, you would have gotten 0 to 0 to 4, and phenotypic also 0 to 4. So that is the end of lesson 4.2. Thank you for watching. Again, really important, make sure that you have your notes, make sure that you wrote down the examples and what you did and that you understand it so that way next class you can complete the practice worksheet. Please reach out with any questions, and thank you again.